Hello, I'm Kevin, graduate student attending Salisbury University's Vascular Biology class. In this video, I'm going to be going through angiogenesis, comparing different models of choice for a hypothetical experiment. The format of the video will follow me giving a little background information on the history on angiogenesis, the two different types of angiogenesis, which are sprouting and splitting, with focus on sprouting angiogenesis, and then comparing in vivo and in vitro assays to fit your potential experimental designs for the future. Angiogenesis is the definition of angiogenesis. Angio means pertaining to blood vessels, and genesis means the formation of something. So angiogenesis is the creation of new blood vessels from existing blood vessels. Before diving into the two different types of angiogenesis, though, I just want to go into a little bit of the timeline of important discoveries bringing us to where we are today in the angiogenesis research. I feel it's important in laying a foundation and appreciating our fellow scientists' hard work and dedication. So, sprouting, which is generally what people think of, if they hear angiogenesis, is the more studied and famous one because we've known about it for over 200 years. Back in 1790, Dr. Hunter was the first recorded evidence of angiogenesis while looking at the inflammation of gunshot wounds. Although he didn't actually call it that, it should be noted he was the first to describe it. It wasn't until almost 200 years later, in the early 1970s, that Dr. Judah Folkman, studying tumors, that the research really began. He hypothesized that tumors are angiogenesis dependent and there was some unknown signaling being relayed to increase the blood supply for tumor growth. So, if you block the signal, the tumor should die. Over 10 years later, two pro-angiogenic factors, VEGF and angiogenin, were discovered, and a few years after that, angiogenesis inhibitors, angiostatin and endostatin, were discovered. So fast forwarding to about 2004, the monoclonal antibody bevacizumab, otherwise known as Avastin, becomes FDA approved for colon cancer and is used for angiogenesis inhibition during research. So there's some key points from history for sprouting angiogenesis, but also important, intussusceptive, also called splitting angiogenesis, was discovered in 1986 by Dr. Bury while studying the microvasculature in postnatal rat lung. For this video, we'll focus on sprouting angiogenesis. And generally, there are four main stages: stimulating the endothelial cells by angiogenic factors, degrading the capillary basal lamina by activating endothelial cells via the extracellular proteinases, capillary sprouting formation and migration of the endothelial cells, and then finally the new vessel magic. Although these four main stages are from a broader spectrum, when reading literature, some researchers might break it into more steps, such as enzymatic degradation of the capillary basement membrane, then endothelial cell proliferation, directed migration of the endothelial cells, tubulogenesis or endothelial cell tube formation, vessel fusion, vessel pruning, and pericyte stabilization. So depending on your study and knowledge at the time of the research, there may be more or maybe even less, but focus on one that may have the closest aspect to your research. So with sprouting angiogenesis, it's essentially initiated in poorly perfused tissues, such as a tumor or just a hypoxic cell in the body. In hypoxia-induced angiogenesis, the parenchymal cell, or the functional cell of the organ, responds to the hypoxic environment by secreting VEGF, specifically VEGF-A. The endothelial cells can then differentiate into two different types of cells, the tip cells and the stalk cells. After differentiation, the endothelial tip cells sprout through the extracellular matrix toward the VEGF stimulus. Tip cells can be characterized by having processes called filopodia that secrete proteolytic enzymes such as matrix metalloproteinases, or MMPs, to help break down the basement membrane of where the angiogenic stimulus is coming from. In addition to the filopodia having proteolytic enzymes, they're highly populated with VEGF receptors, or more specifically the VEGF receptor 2. helps and align more accurately toward the stimulus. The stalk cells behind the tip cells proliferate, causing the capillary sprout to get longer. Once enough filopodia microprojections have reached out to and anchored onto the substratum of the hypoxic cell, actin filaments will align themselves to contract and will literally pull the tip cells toward the stimulus. Simultaneously, though, VEGF-A induced delta-like 4 ligand production by tip cells, which will lead to the activation of Notch receptors in the endothelial cells. The Notch signaling of delta-like 4 ligand mates with its Notch receptor on neighboring cells for cell-to-cell communication and will cause a cascade of events to reach the nuclei of the neighboring endothelial cells, which inhibits them from responding to the VEGF stimulus by stopping production of the VEGF receptor synthesis. The Notch receptor activation will also suppress the VEGF receptor 2 in stalk cells, so only the tip cells continue to proliferate and or migrate towards VEGF. Here, in the stalk cells, vacuoles start to develop and fuse together, which eventually becomes the lumen of the nuke. Keep in mind, this isn't only happening to one endothelial cell. The stimulus was secreted in all directions so they can get a response from wherever possible. So many endothelial cells can be doing this simultaneously until at least 2 sprouts congregate at the source of VEGF. And oxygenated blood reaches the source to decrease the VEGF secretion. In order for the newly formed capillary to mature and stabilize, it recruits pericytes and extracellular matrix formation, as well as mechanical stimulation such as shear stress. I will mention that FLT1 and FLK1 are what VEGF-A interacts with, although the Notch signaling is not fully understood, so I don't want to go into deep details on that and potentially misinform you. So now that we have a basic understanding of angiogenesis, why study angiogenesis? Angiogenesis can be used for many more things to research than just cancer. It's pathologically associated with diabetic retinopathy, some autoimmune disorders, asthma, arthrosclerosis, and many more that I won't name. So, cell culture, or in vitro, human umbilical vein endothelial cells, or HUVECs, are usually used for the most endothelial cell assays in vitro because of how easy it is to harvest them from large vessels. This does bring up a point to mention in which these endothelial cells are not a very good representation of vascular endothelial cells in vivo. A technique called Boyden chamber assay would be a good model for endothelial cell migration. Some other common in vitro assays are the tube formation, tubulogenesis, where you can use HUVECs with Matrigel that represents a 2D assay, or fibrin gel bead assay for a 3D representation. You can use these if you're looking to study one specific step of angiogenesis or some gene expression during that step or something of that nature. Some newer or more practical assays with multiple steps are the embryoid assay or the mouth metatarsal assay, because they have the matrix degradation, migration, proliferation, and tube formation, which represents all the angiogenic functions in an in vitro model. Some in vivo assays are Matrigel plug assay, which might be used for fast screening for inhibitors or stimulators of angiogenesis. The corneal angiogenesis assay, which might be used for a vascular response or finding endothelial markers, although I will say that it's pretty invasive. You also have CAM assays, although there is quite a bit of controversy throughout research for inflammatory response interfering with results, because if you're testing a substance, it gets injected into the eggshell of the chick embryo at like 7 to 9 days. So there's a lot of controversy over that. But that brings us to the hind limb ischemia assay. It's pretty popular for studying peripheral artery disease and vascular remodeling. Of course, all of these in vivo and vitro will have their pros and cons, so it'll be important to determine which one to use to optimize your hypothesis, or funding restrictions, any other limiting factors associated with your scientific study.